Hello, hi, good day, everybody. My name is Natalia Elizalde, Business Development Director at VIVEbiotech, and I'm pleased to have the opportunity to present today at the Cell and Gene Therapy meeting on the Mediterranean. So, in this presentation, I will try to briefly explain who we are, who VIVEbiotech is, and our activities, capacities, capabilities, and so on. So basically, well, we are a company fully focused on lentiviral vectors. We are a GMP CDMO, which means that we are involved in the development of lentiviral vectors from very early stages to the manufacture for preclinical, GMP manufacture for clinical, up to commercial stages within our increased capacity plan that we are completing now. We are indeed continuously working on the optimization of production process, both from the upstream and downstream development point of view. And also, we have an R&D dedicated department whose objective is enhancing the properties of the lentiviral vector itself. We are working globally. Indeed, at the moment, approximately half of our customers are based in the States. And we do have different customers in different countries in Europe, Asia, Israel, Australia, and so on. In this world map, you can see marked in green the different countries where our lentiviral vectors are being used. Okay, we are working in compliance with both EMA and FDA regulations, and our lentiviral vectors are being shipped globally as well. Of course, we are specialized in GMP manufacturing, but we really like to say that we are experts in virology, and that's why we are giving such a great importance to the fact that the collaboration with a given company, with a given customer, should be established the earlier, the better. And that's why we are being involved in processes such as cloning, R&D manufacture, process optimization, and so on. More than 35 different lentiviral vectors have been manufactured by VIVEbiotech. And indeed, at the moment, we've manufactured more than 100 batches in reactors. Which are the applications of the lentiviral vectors we are manufacturing? So approximately half of them are being used for immuno-oncology applications, mainly transducing T cells, but also other kinds of cells such as NK cells, among others. The other half, approximately, is being used for rare diseases by transducing, again, different kinds of cells, such as, for instance, hematopoietic stem cells. And also we are manufacturing lentiviral vectors that are being used, for instance, for in vivo applications or the transduction of iPSCs. Here you can see some of the, collaborations we've established so far, such as, for instance, we are working together with Cyphos, that is an Astellas company, VIVEbiotech based in the States, and also with presence in Israel. And just one acknowledgement that we've been very recently recognized. Now we've been mentioned as the 5th finalist in Spain for the best SME company, and the 26th of February we'll see if we have the privilege of being the ones awarded with this very relevant recognition. So we'll see. What about our team, the people that we form part of VIVEbiotech? So we are continuously growing in a sustained manner. Our headcount is being increased. Here you can see that as a company we were created in 2015. Now we are 63 people, and we expect ending the year in 80 people in the team. So the reason why we are increasing also in terms of the number of people is because we are moving from our current facilities. Here you can see them. These are the labs, this is the offices building, and so on. We are moving to a new building in the same technology park in San Sebastian. Okay? We are based here in the Basque Country in Spain, to this new building where additional 2 rooms are being built. This is important, and it's important because it will allow us, start working new projects, also increasing the batch sizes that we are manufacturing up to commercial stages, and having more flexibility for the projects we are already working in. So last year, we manufactured 20 batches in reactor. This year, we already have the capacity to manufacture 40 batches in reactor. And in 2022, we are already talking about 80 batches in reactor per year. What about the scales? Well, the scales, they are very wide, which is important because they are able to adapt to the requirements that are needed for every developmental phase. For instance, we are able to work from the smallest version of reactor available, 2.4 square meters, or 2 of these in a row, like 4.8 square meters, and so on. These kind of scales, they are very appropriate for developmental phases. Then we also have capacity for 10 square meter batch size, which is very good to complete phase 1 to clinical trials, usually thinking on CAR-Ts and this kind of applications. 30 square meters, 2 of these 30 square meters in a row will be also available, okay, processing this harvest coming from these 60 square meters in a single DSP. And then 200, 600 square meters will be the reactors used up to commercial scale, okay? So these are the batches available now, and from now up to 2023, all these scales will be available. Okay, this is how our process looks like in a simplistic manner, but I think that it could be useful for all of you to have an idea about the approach that we are following. So here, you can see this is the upstream part, and this is the downstream one. For us, both upstream and downstream are very, very important. Obviously, we want to produce, well, a high titer, but also we want to get, at the end of the day, a very pure concentrated sample that, from our point of view, and based on our customers' feedback, has a great impact in terms of the final functionality to transduce the final target cell. So that's why we are also working a lot on downstream and removing impurities, impurines, and so on. So basically, what's made is that hectonin 3T cell bank is the first hit, then it's punished first in flasks and then in cell factories, as well as cell stacks depending on the batch size, and then the reactor is seeded. Okay, the reactor is seeded, and then cell growth continues until day 21 approximately, when transient transfection takes place by using PI as transfection agent, as well as 3 helper plasmids and 1 transfer plasmid. Most of the times we are talking about the manufacture of third-generation lentiviral vectors. Following, the harvest is collected during a process that could vary between 3 to 5 days, or what, it depends. And this crude harvest is processed by a multi-step approach DSP. So at the end of the day, what's got is this concentrated, pure, sterile field lentiviral vector that is being released by our qualified person. Okay. For us, quality is key, and indeed that's why both for non-GMP and GMP manufacturing phases, okay, our quality assurance department is being involved. As mentioned before, we have our own qualified person and our own deputy qualified person, and our GMP certification has been just very, very recently renewed, indeed this month, the 9th of February, not only for API, for like drug substance intermediates, but also for drug products. Very important for the manufacture of lentiviral vectors to be used for in vivo applications. Okay. So just as a summary, we are a company fully focused on lentiviral vectors. VIVEbiotech does not have its own pipeline, which means that we are giving full priority to our customers' projects. We are continuously working on the optimization of both upstream and downstream, and any improvement that is found is then transferred to the service we are offering. So the final aim should be not only getting good titers, but also functional lentiviral vectors, decreasing MOI. The lentiviral vector properties are being continuously enhanced by our R&D department. And our aim is, at the end of the day, adapting to every specific project, okay, in a flexible manner, both from the technical point of view and also from the timelines perspective. So now, well, in the event that you are working in a given project that encompasses the use of lentiviral vectors, and you would like to arrange a meeting together with us, we'll be more than pleased to understand well better your project and to have a dedicated discussion. So thank you very much.